Hello everyone! In this practical, we measure the length and width of leaves collected from the same tree. Then, we calculate the mean, variance, and standard deviation for each set of leaves. Lastly, we plotted a scatter graph to study the relationship between leaf length and leaf width. So, the objectives of this practical are to calculate the mean, variance, and standard deviation, to understand the concept of correlation, to determine the relationship between leaf length and leaf width. So, how is it done? First, we collected 30 leaves from the same tree. Next, the leaves were divided into 3 sets with 10 leaves in each set. We measured the length and width of every leaf using a ruler and recorded the data in tables. Then, we calculated the mean, variance, and standard deviation for each set. Finally, we combined all 30 leaf measurements and plotted a scatter graph to determine the relationship between leaf length and leaf width. This scatter graph shows the relationship between leaf width and leaf length for all 30 leaves. Each point represents one leaf. The graph shows an upward trend, which means there is a positive linear relationship between leaf width and leaf length. This means that as the leaf width increases, the leaf length also tends to increase. Although there are few points that are spread out, the overall trend is still positive. From the graph, we can conclude that there is a positive linear relationship between leaf width and leaf length. This means that wider leaves are generally longer. The calculated means also show slight difference among the 3 sets. Set 1 has the highest average leaf size, while Set 3 has the smallest average. These differences are normal because leaves naturally vary in size, even when they come from the same tree. The variance and standard deviation show how much the measurements differ from the average. A small variance and standard deviation mean the leaf measurements are close to the mean. A larger variance and standard deviation indicate greater variation among the leaves. Overall, the results show that descriptive statistics are useful for describing biological data and understanding natural variation. In conclusion, the objective of this practical was successfully achieved. We calculated the mean, variance, and standard deviation for leaf length and leaf width. The scatter graph shows a positive linear relationship, meaning that wider leaves generally have longer lengths. This practical also helped us understand how descriptive statistics can be used to describe biological data and natural variation. This practical is about the factors that affect the distribution and abundance of organisms in the forest ecosystem. We use the quadrat sampling method to identify different plant species, count the number of individuals, and measure diameter at breast height or DBH. This practical also helps us understand how environmental factors influence where plants grow. The objective of this practical was to introduce the concept of quadrat sampling, improve our observation skills, record the difference in plant distribution, and identify the possible factors that affect the distribution and abundance of plants. The materials used during this practical were raffia string, hammer, vernier caliper, measurement tape, and stick. First thing first, we prepared a 5 meter by 5 meter quadrat in the study area. Next, we identified all 3 species found inside the quadrat. We counted the number of individuals for each species and measured the diameter at breast height or DBH. Finally, all observations were recorded for data analysis. During the practical, we measured and observed the plants carefully without damaging them or disturbing the surrounding habitats. Quadrat sampling allows ecologists to collect reliable data while minimizing the impact on the environment. However, the results only represent a small sampling area and may not fully represent the entire forest. In Quadrant 1, we identified 7 different plant species with a total of 8 individual traits. Lychee chinensis was the most abundant species with 2 individuals, while the other species were represented by 1 individual each. In Quadrant 2, 5 plant species were recorded with a total of 8 individual trees. Hofpia helferi had the highest abundance with 3 individuals, followed by Miletia hemsliana with 2 individuals. The results show that Quadrant 1 contained more plant species than Quadrant 2. However, both quadrats recorded the same total number of individuals. This indicates that species richness was higher in Quadrant 1, while Quadrant 2 had more individuals belonging to fewer species. So, the difference in plant distribution between the two quadrats were likely caused by several environmental factors. These include sunlight, soil moisture, nutrient availability, and competition among plants. Seed dispersal by wind or animals may also affect where plants grow. In addition, natural disturbance and human activities can influence plant abundance. These factors explain why some species were more common in one quadrat than the other. In conclusion, quadrat sampling is an effective method for studying plant distribution and abundance. The results show that different plant species have different distributions even within the same forest. Environmental conditions greatly influence plant growth and survival. Therefore, all the objectives of this practical were successfully achieved. During our visit to TRC-RC, we learned about the importance of forest restoration and biodiversity conservation. We observed how native tree seedlings are grown and cared for before being planted in degraded forests. This helps restore forest ecosystem, protect native species, and support a more sustainable environment. This visit taught me that every tree plays an important role in the ecosystem. Forest conservation requires everyone's support, and planting native trees is one way to protect biodiversity for future generations. The Marine Knowledge Hunt helped us learn about marine biodiversity through fun team activities. We also learned how pollution, especially plastic waste, affect marine life and why protecting our ocean is important for a sustainable future. This activity reminds me that simple actions such as reducing plastics use and keeping our environment clean can help protect marine ecosystem. And please remember, everyone has a role in conserving our oceans. Overall, the field trip and the program helped me understand the importance of biodiversity and environmental conservation. Through the practical activities, nursery tour, and marine knowledge hunt, I gained valuable knowledge and learned that protecting nature is everyone's responsibility. Together, we can create a more sustainable future.